Good day everyone, I am Angelo Dantes Baldidara. So for today's video, I would like to discuss about what is the techniques used in rDNA technology. Techniques used in rDNA technology. So ayan, in the first slide is cloning libraries. So libraries are collection of DNA clones in a certain vector. So libraries are like big storage units that contain a bunch of different DNA clones. So these clones are put into something called a vector. So which helps keep them organized and easy to study. So mag-isip kayo na katulad ito ng bunch of a different puzzle pieces in a one box. So, the library is where scientists can go to find exact piece they need for their research, di ba? Research na naman. So, second is the goal is to have each gene represent in the library at least once. So, once, sabi di ba? Oh, so the objective is to ensure that every gene is included in the library at least once. In the other words, a complete representation of all genes must be present in the library without any exception. So this means that no gene should be left out or excluded from the collection. So the purpose of having a comprehensive library of genes is to facilitate new research and ina-analyze yung genetic information. So which can lead to significant advancement in a various fields. So katulad ng medicine and biotechnology. So next is genomic made from rDNA, fragments of total genomic DNA. So genomic refers to something that is related to the complete set of genetic information found in an organism. So in this context, made from rDNA fragments of total genomic DNA means that a specific type of enzyme called restriction endonuclease, so tinatawag nating RA, was used to cut all the genetic material DNA from an organism into smaller pieces. From these smaller pieces, a subset was selected and assembled together to create what is known as genomic DNA. So this process allows researchers to study specific regions or genes within an organism's entire genome without having to analyze the entire genome at once. So next is cDNA complementary DNA made from DNA synthesized from rmRNA. So cDNA, which stands for complementary DNA. So ito ay type of ng genetic material that is made from mRNA. So ito yung process na ito, involved siya sa synthesizing DNA. Ginagamit niya yung RNA molecules as a template. So essentially, cDNA serves as a copy of the original RNA sequence and can be used to study a gene expression or create recombinant proteins. So ito ay importante na maalam natin na yung cDNA only contains coding regions and lacks introns or non-coding sequences found in genomic DNA. So overall, cDNA allows researchers to better understand na kung paano yung genes are regulate and expressed in different tissues or conditions. So this is example of cloning libraries. So in the next slide is the polymerase chain reaction, or we call is the PCR. So allows the isolation of a specific segment of DNA from a small DNA or cell sample using DNA primers at the ends of the segment of interest. So this text is talking about the process called DNA isolation. So ito ay involved sa pagkuha ng small sample ng DNA or cells na ginagamit yung something called na DNA primers to para i-isolate niya yung specific segment of the DNA. So the DNA primers are located at the ends of the segment that researchers want to study. So think of it like trying to find a needle in a haystack. So the specific segment of DNA is like the needle. So and the rest of the DNA in the sample is like hay. So, by using these special primers, so yung mga scientists uh, can pull out just what they need. So, that one little piece they interest in studying. So, overall, this technique allows researchers to zoom in on very specific parts of our genetic code and learn more about how genes work and how they might be linked to different diseases or conditions. So this is the example of polymerase chain reaction. So, so number one stands for denaturation. So number two is annealing. So number three is elongation. So in the next slide is what we call the gel electrophoresis. So DNA fragments of different sizes can be separated by an electrical field applied to a gel. So the negatively charged DNA migrates away from the negative electrode and to the positive electrode. So the smaller the fragment, the faster it migrates. So have you ever heard of DNA? So it is like a blueprint of for living things. So scientists can study DNA to learn more about different organisms and how they work. So but sometimes they need to separate the DNA into smaller species so they can study it better. That's where an electrical field and a gel come in. So the gel is kind of like a jello pero maliit siya or but thicker. So, pero yung mga scientists, nilalagyan nito ng DNA sa gel and ina-apply niya yung electrical field. Something interest happens. So, the negatively charged DNA moves away from the negative electrode. So, katulad ng isang magnet, nilalayo niya to another magnet with the same charge and towards the positive electrode the opposite end. So but here's where it gets really cool. So smaller fragments of DNA move faster than bigger ones. So by using this technique, scientists can be separate out of all different sizes of DNA fragments and pinag-aaralan nila individually. So this helps them understand more about how organism works at a molecular level. So there you have it. So separating out bits of genetic material using electricity. Ito naman yung process ng gel electrophoresis, no? So next slide. So restriction enzyme mapping. So frequently it is important to have a restriction enzyme site map of a cloned gene for further manipulations of the gene. So this is accomplished by digestions of a gene singly with several enzymes and then in combination. So dito, yung mga scientists, gusto nilang manipulate yung clone gene. So it is open para matulungan niya for them to have a map of the restriction enzyme sites on that gene. So this is done by breaking down the gene using different enzymes both individually and in combinations, no? Nagets po ba? So by doing this, researchers can get a better understanding of how the gene works and what they need to do in order to modify it. So essentially, it is like creating a blueprint or a roadmap for the gene so that scientists know exactly where they need to make changes. So next is the fragments are subject to gel electrophoresis. To separate the fragments by size and the sites are deduced based on the sizes of the fragments. So, yung mga scientists naman, pinag-aaral nila yung DNA. So, they sometimes need to figure out the order of the letters so that make up the genetic code. So, to do this, they break apart the DNA into smaller pieces. So, tinatawag itong fragments and then ginagamit nila yung process na tinatawag na gel electrophoresis. So, para ma-separate yung fragments based on their size. So, by looking at how big each fragment is, so yung mga scientists can figure out where it came from in the original DNA sequence. So, isipin mo yung katulad ito ng isang sorting puzzle pieces by size to help put together in a picture. Okay, nagigets pa po ba? Ayan. So, dumako naman tayo sa nucleic acid hybridization. So, a Southern allows the detection of a gene of interest by probing DNA fragments that have been separated by electrophoresis with a labeled probe. So, in the northern blot, probed RNA on a gel with a DNA probe. So, in the western blot, probe proteins on a gel with an antibody. So, the given text ay talking about three different techniques that are used to detect specific molecules in a sample. So, these techniques are called southern blot, northern blot, and western blot. So, in the southern blot, ginagamit naman para ma-detect nyo yung particular gene of interest in a DNA fragment. So, this technique involves separating the DNA fragments using electrophoresis, and then ginagamit nyo yung pag-label ng probe to identify the specific gene you're looking for, no? So, in the northern blot, is similar to southern blot naman siya. So, but it is used to detect RNA instead of DNA. So, in this technique, you use DNA probe on an RNA gel-like gel pala, no, to find the specific RNA sequence you're interested in. So, finally, Western blot is used to identify proteins in a sample. So, itong technique na ito, nakapaloob dito yung pag-separate ng proteins on a gel and then using an antibody as a probe. So, the antibody will bind specifically with the protein you're trying to detect so that you can use it to on the gel. So, overall, these three techniques are very useful tools for scientists who want to study specific molecules like genes, RNAs, or proteins. Okay? Okay. So, ito naman yung process ng nucleic acid hybridization. So, dumako naman po tayo sa DNA microarrays. So, vast majority of the protein encoding qualities onto microarray chip. So, utilizing innovation in light of the DNA silicon chip industry. So, the chip can be utilized to hybridize to cell RNA and to measure the statement rates of a substantial number of the qualities in a cell. So, yung text na to ay inaalam niya about sa new technology na ginagamit yung microarray chips to measure the expression of level of many genes in a cell. So, these chips are made of using techniques from the DNA silicon chip industry and kinocontain yung protein encoding qualities. So, the chip is able to bind with RNA from cells and determine how much each gene is being expressed, so, or spoken, no? So, in that cell, no? So, this allows scientists to study many genes at once and magain yung mga insights sa kung paano mag-work together in a different biological processes. So, ito naman po yung techniques na ginagamit sa DNA microarrays. So, ayan, dumako naman po tayo sa DNA sequencing. So, DNA sequencing refers to the general laboratory technique for determining the exact sequence of nucleotides or bases in a DNA molecule. So, the sequence of the bases often referred to by the first letters of their chemical names. So, the A, T, C, and G. So, napag-aral naman po natin kanina, di ba? So, encodes the biological information that cells use to develop and operate. So, DNA sequencing, ito yung method na ginagamit niya sa laboratories para mahanap yung exact order ng building blocks. So, tinatawag natin itong nucleotides or bases in a DNA molecule. So, these bases are usually represent by the letters A, T, C, and G. So the sequence of these letters contains important information that cells use to grow and function properly. Ayan, dumako naman po tayo kay Erwin Chargaff's rule. So, also known as the complementary base pairing rule. So, it states that DNA base pairs are always adenine with thymine, so or the AT, no? And cytosine with guanine, C to G. So, a purine always pairs with the pyrimidine and vice versa. However, A doesn't pair with C despite that being purine and pyrimidine. So, Erwin Chargaff's rule is a scientific concept that explains how DNA-based pairs always match up in a specific way, no? This matching is called complementary base pairing, and it means that adenine A always pairs with thymine, letter T, with cytosine C always pairs with guanine, letter G, no? It is important to note that purines, which are larger molecules, ano po, no? Tandaan nyo yun, will only pair with pyrimidines, no? Which are smaller molecules. So, these rules helps ensure the stability of the DNA molecule, no? Okay? So, however, there is a exemption to this rule. So, does not pair with C even though they are both purines or pyrimidines. So, while it may seem strange at first glance, so this actually makes sense because A to C pairing together would result is unstable hydrogen bonds between the two bases. So, overall, Erwin Chargaff's rules help scientists understand more about sa kung paano yung DNA works and why certain base pairs are necessary for its structure and function. Okay, nagets po ba? So, this is the rating complementary DNA sequences. So, A to T, C to G. So, A, G, C, T, G, G, T, 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 G, A, C, G, A, C, down to T, T, C, G, A, C, C, A, C, T, G, C, T, G. Ano po? So, next example naman po is yung A to T, C to G. So, A, T, A, C, G, T, G, G, A, T, G, C, T, A. Down to T, A, T, T, G, C, A, C, C, T, A, C, G, A, T. So, another example na naman po ito. Kayo na pong bahala magbasa dyan. Okay. So, we're going to talk about chemical cleavage method by Alan Maxam and Walter Gilbert, 1976 to 1977. So, Maxam-Gilbert sequencing was the first widely used method for DNA sequencing. So, it involves modification of the bases in DNA followed by chemical base-specific cleavage. So, the chemical cleavage method, also known as the Maxam-Gilbert sequencing. So, ito ay technique na ginagamit to ma-determine yung order ng DNA bases. So, this method was developed by two scientists named Alan Maxam and Walter Gilbert in 1976 to 1977, no? So, ito ay naging popular for DNA sequencing, ano po? So, the process involves changing the chemical structure of the DNA bases and then breaking them apart into smaller pieces using specific chemicals that target each base individually. So, by analyzing these small fragments, so yung mga scientists, dinidefine nila yung sequence of bases in a segment of DNA. So, this method was important, ano po? So, to break through in genetic research because, it allowed scientists to study genes more closely and understand how they work. Ayan. Ayan, so we're going to tackle about the sequences DNA fragments containing up to 500 nucleotides in length. So, itong method nito, ginagamit yung double-stranded DNA sample. So, bases of DNA molecule are selectively destroyed, not used very much anymore because reagents are highly toxic and very dangerous. So, this text about tinatawag natin itong sequencing which is used to study DNA fragments that are made of to 500 nucleotides long. So this method ni-requires niya yung double-stranded DNA sample. So sa prosesong ito, specific parts of the DNA molecule ay kayang sinisira or nire-remove selectively. However, this technique is not commonly used anymore dahil ito ay involved sa highly toxic and dangerous chemicals. Ayan. So, para mas malaman nyo ng maigi, no, sa sequencing, tinutulungan niya para maintindihan natin yung genetic makeup of living sequences. By studying the order of nucleotides, so yung A, T, C, and G in a fragment of DNA, so yung mga scientists, dinidefine nila yung important information katulad ng organism's traits para ma-certain yung diseases. So, in this particular method mentioned in the given text, so, only short fragments containing up to 500 nucleotides lamang siya can be studied at once, no? Once. To do so, yung double-stranded DNA samples, so, ni-require niya para ma-provide yung two complementary strands for analysis. So, however, one's drawback of this method is that is used reagents, so yung original chemicals na highly toxic and dangerous when handled improperly. So, kailangan dapat maging maingat tayo sa paggamit doon. So, these reagents selectively destroy some parts of the DNA molecule while leaving others intact for analysis. So, due to safety concerns associated with these chemicals and better alternatives available today, so this technique is not widely used anymore. Ano po? So overall, sequencing plays a crucial role in modern biology research and has revolutionized our understanding of genetics over time. So okay, nagigets pa po ba? So, ayan, to po yung last topic ko. So, yung the enzymatic method or the Sanger-Dioxy or the primary extension chain termination method. So, DNA sequencing, so the enzymatic procedure is commonly referred to as Sanger-Colson method since it was developed by F. Sanger and the coworkers. So, the enzymatic method, ito ay tinatawag din Sanger-Dioxy or the primary extension chain termination method. So, ito yung process na ginagamit sa sequence ng DNA. So, itong technique na ito was developed by F. Sanger and his coworkers. And it's often referred as the Sanger-Colson method. So, during this procedure, enzymes are ginagamit nila to extend yung primer that binds to a specific region on the DNA strand. So, the primer acts as a starting point, no, for the sequencing reaction. So, once na dinagdagan ito, so yung small amounts of the nucleotides, the building blocks of DNA are introduced into reaction mixture. So, sa process na ito, minamodify niyo yung nucleotides na tinatawag natin nucleotides or the DDNTPS, small letter D, small letter D, big letter N, big letter P, T pala, and big letter P and small letter S. So, are included into low concentrations along with the regular nucleotides or the DNTPS or the small letter D, big letter N, big letter T, big letter P, and small letter S. So, this DDNTPS lack of an essential chemical group required para malaman yung extension of the growing chain once they have been incorporated into it. So at each DDNTP is added during synthesis through complementary base pairing with these template strands at their respective positions in a relation to primers. So polymerase activity stops because there is no 3 negative OH group available for continuation of elongation from which post-polyester bonds can be formed between adjacent monomers. So, ayan, nagiging biochemistry na tayo. So, this leads to chain determination since no more bases can be added beyond that point due to lacking necessary groups needed for linking units together via covalent bond formation. So, yung resulta ng fragments differ in length based on where these terminations occur relative position wise within sequences. So, being read out using yung mga fluorescent labels attached onto them afterward. So, they may be detected by automate machines capable of reading out such as signals generated upon excitation by laser beams. Ayan, layo na natin. So, overall, this enzymatic method provides sa mga researchers para ma-accurate yung information about the genetic material at para matulungan yung mga scientists para kung paano gumagana yung genes together within an organisms. So, in the second to the last, Sanger sequencing is a method of DNA sequencing based on the selective incorporation of chain terminating dideoxy nucleotides by DNA polymerase during in vitro DNA replication. So, Sanger sequencing, ito ay paraan para ma-figure out yung order ng sequence of DNA, no? So, it works by using a special kind of molecule na tinatawag nating dideoxynucleotides. So, to stop the replication process at a specific point. So, ito ay inaalam nyo ng mga scientists para mabasa at ma-record yung base pair in the DNA sequence. So, itong method na ito is often used in the research labs and medical settings para mapag-aralan yung genetic diseases and para malaman yung mga mutations that may cause them. Okay? So, okay pa ba kayo? Ayan, no? So, last topic na ko na, no? Ayan. So, the classical chain termination method requires number 1, a single-stranded DNA template. Number 2, a DNA polymerase. Number 3, normal dideoxynucleotide. The triphosphates DDNTPS, yung sinabi ko kanina. So, the classical chain termination method is a technique used to sequence DNA. So, it requires niya yung 3 main components. So, a single-stranded DNA template, a DNA polymers, and normal dideoxynucleotide triphosphates or the DDNTPS. So, in a single-stranded DNA template, ito ay is the starting material for sequencing. So, ibig sabihin, yung strand of DNA being sequenced has been separate from its complementary strand. So, that it can be read one base at a time. So, DNA polymerase is an enzyme para matulungan niya ma-build yung new strands of DNA para madagdagan yung nucleotides in the correct order. So, in this case, it will add either normal nucleotides or NTPS depende on kung ano yung type of base needs to be add next, no? So, normal deoxynucleotide triphosphates are special types of nucleotides that contain fluorescent tags. So, when they are incorporated into growing strand of DNA, so ito ay nagkakos ng chain termination dahil nalak nyo yung necessary of chemical group to form another bond with other bases. So, by using these three components together in a specific way, no? So, yung mga scientists, nababasa nila yung sequence of bases in a given piece of single-stranded DNA. So, yung process, naka-involve dito yung adding each individual base sequentially and observing which DDNTP. So, nagkakos ng chain termination at each step. So, this information, ina-allows niya to determine which base was present at each position along the original strand. So, ayun lang po sa aking part. Sana marami po kayong natutunan. Maraming salamat po. God bless you.